Well, thank you everyone for joining. My name is Kelsey Achenbach. I'm the head of pharmaceutical marketing for Roquette. And today I'm going to talk about a couple different things. I'll talk a little bit about Roquette, but this is not a presentation about Roquette. This is actually a presentation that I want to start the discussion around the relationship between raw material suppliers and biopharmaceutical companies. So I don't think we talk about that enough. So that's where I'm going to maybe plant some seeds into the audience today and hopefully start the discussion. So first of all, a little bit about Roquette. So we are a family-owned French company. We have over 20 manufacturing facilities and operate in over 100 countries. For pharmaceuticals, for small molecule pharmaceuticals, we have a wide range of excipients that are seen in almost every dosage form. For large molecules, we have ingredients that are both in cell culture solutions and protein stabilization, so both upstream and downstream. You can see we have over 300 people in our workforce in R&D. So we're really focused on understanding the science behind our materials and, like we like to say it, unlocking the potential of nature. But that's not what I'm here to talk about today. Today I want to talk about the monoclonal, the history of biologics, and specifically where we've been and where we're going for monoclonal antibodies. So arguably you can say that biologics started a century ago when we first started looking at vaccines in the early 1900s. Really, there wasn't the first hint of monoclonal antibodies until DNA was discovered in the early 1950s. And it really didn't come into fruition until those early stage companies in the 1980s. The Amgens, the Genentechs, they pioneered the entire industry. They really, when they started as startups and created that startup mentality in the pharmaceutical industry. But today, biopharma is not a startup. It is well-established industry, and does that help? It is a well-established industry, and the landscape is changing. Tomorrow, it's probably going to cell and gene therapies. We heard about that a little bit earlier today in some of the discussions, and you know, in 2018. There's over 150 INDs that were submitted into CBER for cell and gene therapy. So that's the next generation. But monoclonal antibodies, they're kind of maturing as a market. So let's talk about that. How is that maturing, and what does that mean for raw material suppliers? So really, monoclonal antibodies established themselves in the industry. The key to their success has been the ability to develop robust manufacturing processes. And that's kind of lined out for monoclonal antibodies. I heard in one of the presentations earlier today that the process is now easy for monoclonal antibodies. And I thought that was an interesting comment, because when it started, it wasn't, right? It took us a long time to get there, but it's there. And now with biosimilars, now is really the time to focus on that processing stage and the optimization of that process, and really look at how do we improve the yields, which a lot of work has been done on, and lower the cost of production. And when you're looking at improving yields, you're obviously focusing on titers. Titers have increased, the average titer has increased significantly over the years. You know, you can see from the 1980s, less than half a gram per liter. Now the average is over 5. So it's a significant increase. A lot of that can be attributed to process optimization. But it's not just process optimization that comes into play when you're looking at increasing titers. It's everything around the cell. So if you think about it, we're taking a cell and we're putting it into an alien environment, right? A bioreactor. There's a lot of different things that can happen to the cell that can affect whether or not your cell is going to continue to multiply. And you can look at molecular design, environmental design, the media design, and a lot of work has been done around media designs. I really hand it to the industry. The media formulation has really optimized the titer yields. But what I see hasn't been done is a lot of focus on the raw materials that go into the media. There's still a lot of work to be done, and more understanding of how we can optimize by understanding the raw materials in the media selection. So I'd like to give you an example, a little mini case study. So Roquette is a manufacturer of dextrose. We manufacture both anhydrous and monohydrate dextrose. It's a picture of a manufacturing facility in Le Trem in northern France. We were working with a customer that wanted to change their supplier of dextrose. And they wanted to for a different reason. It wasn't to increase yields. They changed over to our source of dextrose. And they increased a tighter yield of, they achieved a 20% increase of their titers in their yield. Significant, massive. They were surprised. We were surprised. So if you look at the dextrose side by side with the competitor, the COAs look exactly the same. So you would think that dextrose is dextrose, but it's not. So we're currently working collaboratively with this organization to determine what is going on here. We don't have all the answers today, but we do have some ideas. So it could be the impurity profile. It could be heavy metals inherently in the dextrose from the raw material, and we know heavy metals can have an impact on cells. It could be the purification processes that Roquette uses versus other dextrose suppliers. We don't know. This is exactly the reason that we opened a biopharmaceutical laboratory in our Asian headquarters in Singapore last year, to understand some of these impacts of raw materials on cell lines and specifically on biopharmaceutical manufacturing. There's not enough work that has been done there today in the industry. So this is a great example. This is an example for upstream. We also have some examples for downstream processing. So that's really where I wanted to push the thought process today, and I want us to start that discussion of how do we collaborate more between raw material suppliers and biopharmaceutical manufacturers to understand these kinds of impacts on the raw material, even if they look the same side by side. So a lot of that work has been done in small molecules. And if you think about CPhI, this is a place where small molecules and large molecules are starting to combine. There's a lot of work that have been done in small molecules around QBD. And if you talk to me as a small molecule manufacturer, and I could tell you, well, my scientists can tell you that this raw material change in our process or this morphology of our product can affect your drug this way. We know that. The excipient suppliers know that. We're not there yet with large molecule. There's a lot to learn and a lot for us to discover. And we need to discover that together and collaboratively with the biopharmaceutical manufacturers. So if you look at your process development stage, you're always aligning it to your clinical development phases. Where QBD starts is right around here. And that's where you should be engaging raw material suppliers. And if you look, there's a study that was done, and QBD has been looked at quite a bit in biopharma. There's a study that was done, but it hasn't been implemented very well yet. In 2008, late 2009, there's this collaborative effort, a CMC biotech study done with AbbVie, Genentech, Pfizer. There's about 6 GSK. There's about 6 different companies there. They worked together to develop kind of a roadmap on what would QBD look like for biopharma. And a lot of the focus was on, as it should be, right, on process development, understanding the different process parameters and the effects on the drug product. There's only a limited work done on the raw material. And most of that work you can see was done based off from prior knowledge of raw material. So my question to you and to the industry is, what if that prior knowledge doesn't exist? Because there's still a lot of work to be done on how these the changes in raw materials affect each parameter of the process. So just relying on prior knowledge doesn't make sense. We need to understand how these different products affect cells in particularly. So that's the seeds I wanted to plant for you today. I believe there's a lot more work to be done in collaboration of raw material suppliers and biopharmaceutical manufacturers, and that's where we're going. We've invested in a laboratory to understand just that, and we'll continue that process. So I'm open for any questions.
Thank you very much. As you just said there, Kelsey, if there are any questions, please raise your hand. I have a quick question for
Sure.
you. Obviously, Roquette, you supply raw materials to both the small molecule and the large molecule firms. Can you just talk us very briefly through a couple of the differences that you've seen from the biologics industry as to their demands there?
Yes. I mean, the biologics industry, it is demanding. I don't know. I would say that the demands aren't that much different, right? The demands is you need high quality. There are some specific demands for biologics like fiber free, but we're seeing that come over to small molecule injectables. That's there. What I would say is in biopharma, it's strange. It's a little bit strange for me. What I see is that most raw materials change hands 2, 3, 4, maybe 5 times. And so there hasn't been a lot of focus on understanding where those raw materials come from, just that they're coming to you in the right way and they're packaged in the right way. The a lot of the chemicals that are used in the biopharma industry are industrial chemicals, right? So there's variability there that we don't yet understand that I think we're going to start to see, especially as the monoclonal antibody market is maturing and biosimilars come in. There's going to be a lot more work done on that area.